Your cells are constantly taking in nutrients and removing waste. Let's discover how things enter and leave the cell. Imagine a cell as a tiny living city. It constantly needs nutrients, water, and ions. But how do these substances cross into and out of the cell?
The plasma membrane is mainly made of a phospholipid bilayer. The hydrophilic heads interact with water, while the hydrophobic tails
Form a water-repelling interior.
The first mechanism is simple diffusion. Molecules are constantly moving because of thermal energy.
Small nonpolar molecules such as oxygen can cross directly through the lipid bilayer. But not every molecule can pass through the lipid bilayer. Glucose and many ions are too large, polar, or charged to cross the hydrophobic membrane easily. Osmosis is a special case of passive transport involving water. Water crosses through specialized membrane proteins called aquaporins. Movement depends on conditions. Sometimes the cell needs to move a substance in the opposite direction from its concentration gradient. Transport proteins cellular energy, commonly from ATP, to moves against their gradient. What happens when the material is too large to pass through a membrane protein?
The cell can use the membrane then pinches off, creating a vesicle inside the cell.
The opposite process is exocytosis. Here, an intracellular vesicle moves toward the plasma membrane and fuses with it. The vesicle releases its contents outside the cell.
So, how does a cell control what enters and leaves? Small nonpolar molecules can cross by simple diffusion, and very large materials leave through exocytosis. Diffusion is the easiest way to remember them. Diffusion moves down the gradient. Facilitated diffusion needs a protein but no direct ATP. Endocytosis sends material.
Quick question.
Which transport mechanism requires ATP to move substances against their concentration gradient?
The answer is C, active transport.